Assalamualaikum warahmatullahi wabarakatuh. Hi class, glad to see you again. So we are now continue on chapter 10 for ART, Assisted Reproductive Technology. So I want you to recall back what we have learned before in the class. First one, to discuss on what are the infertility factors among men and women, and second one, what are the types of assisted reproductive technology are there. So we have learned about IVF, or rather than we just know IVF in human, so we do have IVF in animal, that actually the manipulation for embryo to be easily fertilized inside the animal itself. So animal research, for example, for the cow, sheep, and so on. So these are the one, once they collect the oocytes and fertilize and culture, this one is to ensure the quality of the eggs and so on. So the one embryo itself will be transferred to another kinds of species. So breeding based on the client needs on how what are the quality that they want. So the potential outcomes of IVF inside the animals to produce offsprings that are having terminal illness means once they got into the disease, they can cause the death. So cause death, this is what we call as terminal illness. And then to produce offspring from cows that are infertile because of the old age or disease, as well as the cystic ovaries. So this one, the culture, they improve the heart quality, and result the calf being born earlier, and then they are having higher or much more quality rather than previous parents, and increase the reproductive performance. Okay. So use the IVF cloning, one types of crucial part to know about the clone. They also using the same technique of IVF, but cloning what we call as like photostat. Okay, photostat is like to get the same organisms, means copycat. The same copy, that's what we call as cloning. But there are many legal and ethical issues surrounding to use in spite of their advanced reproductive techniques. So we do have copy the same genetic makeup as the original biological identity. This is what we call as cloning. So they allows to produce offspring that genetically identical to the single donor of the animals. And we do have one technique, what we call as somatic cell nuclear transfer. Okay, this is where the fusion of the nuclei are being exposed entirely, means nucleus from the female and male to got diploid donor cells with unfertilized or unnucleated oocytes. So this is the first one, the first success cloning inside Dolly, one sheep, mammal sheep that been cloned, the first one that are successful. They create new organism. Okay, the organism to be cloned, they ambil satu part of it, the developed differentiated cells taken from the organisms. Then how is it? Dia akan ambil unfertilized egg cells, and then nucleus are being removed from the egg cells from another organisms. And then egg cell with no nucleus are being inserted with the one that we want to clone. So then they are fused together, then the development as embryo is being assessed, then they are implanted into a new rumah yang baru lah. So this is what we call as the clone will be born. So ni adalah cara untuk dapatkan clone bagi this organism. Tapi diperlukan another organism yang dibuang nukleus dia. That's why we call as somatic cell nuclear transfer. Okay, they are unfertilized, coupled with unfertilized, unnucleated oocyte. So for agriculture, they are mostly applied. Okay, maksudnya digunakan secara meluas. Also for bacteria, they are extracting the DNA isolation, the clone of the gene are being are being copied many times, and they are transformed into cell and tissue culture. So inside the lab itself, they are being developed to become a full functioning plant. So this one mostly to give a rapid distribution of the genetics, for the genetics improvement, and to conserve the genetics itself. Okay, so this is technology that isolate particular gene. This one, what I call the particular gene are being isolated from organism that we target, and then from the gene itself, they give a information of genetics will be produced from RNA and protein molecules. Maksudnya akan ada proses translation and transcription that will then transcribe into the protein that will successfully akan produce that things. So the modification existing organisms so that they express a trait not previously encoded in the genome, and this one what we call as transformation. Maksudnya daripada organism, contohlah bentuk dia circular, kita ambil gene yang ada bentuknya triangle, then they will be transformed into this kind of phenotype. So this is the simpler way lah I want to see that a trait not previously encoded the genome. So this is what we call as transformation. Transform. So we do have a lot of types of cloning, from gene cloning, reproductive cloning, and therapeutic cloning. What are the differences between them? Gene cloning, they are having the DNA fragment from one organism. So daripada DNA fragment, they will be self-replicate to, for example, bacteria plasmid. For reproductive cloning, dia akan ambil copies from the organism, identical copies, tapi using their own DNA, but non-sexual means. Maksudnya bukan daripada DNA yang target for sex DNA, not from sexual chromosome. Okay, so daripada copies of an organism can create identical twin and born years later. Okay, then we do have therapeutic cloning. that produce identical copy of an organism cells using its own DNA, and then can be potentially used to replace diseased organs and cancerous tissue. Okay. This one, the most unethical things to try to clone a human. Okay, so this one is macam dibincangkan lah. Can we clone a human? Okay, so basically the process is the same. We isolate the cells, and then we try to have another ovum from female. So do the isolated somatic cells will be, okay, this nucleus will be removed and being insert with the isolated somatic cells, and then the eggs with somatic nucleus will be activated by the shock, and you can see early embryo development in the lab, and then they will be inserted into host of the mom and got the clone body. But is it ethical? So this ethical consideration should be this one, what we call as the embryonic stem cells of human. Is it ethical? But mostly based on the religion perspectives, is unethical, and it is banned lah, macam tak boleh lah nak buat clone of human. Can we do have stem cells technology. Okay, stem cells technology, unspecialized cells maksudnya cells yang masih tidak berdivided akan digunakan dalam ni. Kita akan pilih satu. Then they can renew themselves by division of mitosis for long period. Under certain condition, can be induced to become cells with specific function. For example, these cells can be developed into heart or muscle cells or lymphocytes. Okay, so this is what we call as embryonic stem cells and adult stem cells. Then this stem cell can be clone of adult cells, more commonly produced in vitro fertilization, and they are broadly characterized into multipotent stem cells or pluripotent stem cells. Okay, apa beza pluripotent stem cells dengan multipotent? Pluripotent, they are capable giving rise to most tissue of an organism. But for multipotent stem cells, only specific function. Cells with only specific function, for example, blood stem cells hanya boleh jadi blood, not another types. But for pluripotent, it can develop into a lot of functioning tissues such as muscles or heart cells and so on. So this is the differences between pluripotent and multipotent stem cells. And we do have embryonic stem cells. Okay, the one that unspecialized or undifferentiated cells that can be potentially developed into any kind of cells. Okay, this one are being studied very well now. By 2026, it's now being studied, but it's not inside the human, but inside the animals. Okay, embryonic stem cells are not harvested from mother or embryo, but they are created in vitro. By the base are inside the laboratory, or what we call as cell culture. Okay, the cells that are being used are at blastocyst stage. Maksudnya stage yang dalam nilainya 30 sel saja yang masih baru develop, tapi they are produce the same IVF, tapi embryonic stem cells are not implanted inside the uterus. Okay, kalau dia implanted inside the uterus, this one untuk IVF, untuk dilahirkan baby tu. Tapi untuk embryonic stem cells, dia hanya berada di blastocyst stage, and they are maintained undifferentiated over long periods. Maksudnya dia disimpan ataupun cryopreserve. What we call as cryopreserve. Okay, so they can be mixed with chemicals to help the cells to differentiate into any different properties. So actually, this one is one of the means to treatment for chronic diseases through therapeutic cloning. So this one can form two healthy offspring. Okay, then we do have another one, embryonic stem cells, what we call as totipotent. Totipotent, this one, totipotent stem cells. They are having unlimited capability to form extra embryonic membranes and tissue and embryo itself. So what we call as totipotent. They are fully capable of giving rise to an intact embryo. So this one, the successful rate nowadays is lesser rather than multipotent ataupun pluripotent stem cells. Okay, this is how we want to generate embryonic stem cells. Once the sperm and egg are joined, they are incubated to develop in 5 to 7 days to produce blastocysts. Blastocysts do have 30 cells and so on. So this one occur inside the laboratory environment. So the blastocyst will be removed its inner cell mass. Okay, then they will be grow in a petri dish in vitro. Okay, so the change of culture condition, maksudnya dia akan letakkan chemical yang berbeza to stimulate what type of cell that you want to differentiate. For example, if you want to develop it into neural cells, boleh. Into skeletal muscle cells pun boleh. Into skin cells pun boleh. So untuk apa all of the cells? Untuk gunakan untuk treatment for chronic disease. So that's why we are using these embryonic stem cells. Okay, we do have pluripotent. Okay, pluripotent stem cells that only ability they can differentiate into certain tissue only. Okay, for example, blood stem cell can develop into RBC but not muscle cells and can be extracted from adult. For example, bone marrow sample. Daripada bone marrow ni kita akan ambil isolate contoh daripada patient dan grow inside the dish, treat cells with programming factors, wait for few weeks. Dia akan tengok daripada tisu, dia akan going to change the culture condition, dia akan stimulate untuk keluarkan differentiate into a variety of cell types. Okay, so these are the location for somatic stem cells inside the body. These are the possible tissue that can be differentiated inside the lab. Then we do have multipotent stem cells that demonstrate only the ability to self-renew or differentiate into specialized types sahaja, tanpa jenis yang lain. For example, hematopoietic only into blood cells, mesenchyma into the one that are related to the osteoblasts, myocytes, chondrocytes, and adipocytes. For neural stem cell can be differentiated into neurons, astrocytes, and oligodendrocytes. What are the key features? They only present is small in number. Primary function to replenish damaged or apoptotic cells. They are remain present, maksudnya dormant until they perceive signals to differentiate. So this one we can give the initiation to inside the lab. Maksudnya kita akan letak chemical ataupun media and so on. That's to activate it to be inside a specific type of cells. Okay, for example, all bloods are related for multipotent stem cells. Okay, this one specialized Specialized, okay, specialized myeloid stem cell boleh give rise into sama ada sel darah merah, sel darah putih, this one, ataupun thrombocytes untuk faktor bagi darah untuk membeku. Or we do have lymphoid stem cells to produce B lymphocyte ataupun T lymphocyte untuk secondary immune response. What are the potential benefits of stem cell research? We can understand how the development works, maksudnya development of an individuals. Daripada satu tisu, daripada satu cells menjadi tisu dan develop into organ. So we can learn and understand how it is work. And how the evolution also daripada dulu kenapa kita ada what we call as perbezaan dan kenapa ada types of inheritance yang berbeza. So these are benefits untuk stem cell research. Diagnose and remedy birth defects, replace damaged disease of organ, help to cure the cancer, and start to take the case for testing a new drugs. Maksudnya kat sini tak perlu dah guna manusia untuk test drugs, tapi kita boleh guna sel manusia yang di-grow dalam lab. That's why we call as substitute the case for new testing drugs. Okay, that's all for chapter 10, and then stay tuned for chapter 11 and 12. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.